Okay, all right. Hey guys. So I hope you guys enjoyed at least one of the papers, because I know the second one, the social one, was definitely, I think, more interesting and pertaining to us a little bit more. But I also did enjoy this first paper. Credit to you. So thank you. Okay. So opioids, right? So does anybody want to explain to me or what they know about opioids in the first place?
Call on somebody. Jake.
Go ahead.
Hurry up.
Well, it's just like a lot of it's like pain relievers like out there, but
yeah.
And they're also like really addictive, so that's why it's like
yeah,
it's a big issue.
Exactly. So they're a common pain reliever, right? So a lot of people who go through acute sensitive pain or like after surgery or some sort of injury, doctors prescribe opioids to help relieve the pain, just so that they can get some sort of relief. So they have a proper use to them, but it's just that they can be highly addictive, as you said, and that's one of the biggest problems that happens with opioids and inside of like pretty much across the world. So so there's a basic mechanism that works for opioids, and so basically what happens is we have in our brains and in our neurons we have these things called opioid receptors, and so basically what they do is that we will have some sort of endorphin or some sort of opioid that attaches to these receptors, and it's an inhibitory neuron, which means that when a receptor binds to it, it causes more neurotransmitters to be released, and when they attach to these receptors, they inhibit the release of dopamine. So pretty much when it's not present, when the opioids are not present, it will release this neurotransmitter, causing it to attach to this close by receptor right here, inhibiting the release of dopamine. But what happens when you do have opioids or some sort of endorphin or something along those lines, it will attach to these receptors right here, which inhibits the release of this neurotransmitter, and it doesn't bind to these receptors right here, which allows dopamine to be released. Now, I hope that makes sense. I know it's a little confusing, but that's pretty much what it does. And so when we take in opioids, it releases a lot of dopamine, which is the neurotransmitter that signals happiness or euphoria, something along those lines, right? And so a lot of people, especially depending on where in the brain that dopamine is released, will either cause you to feel intense sensation of euphoria and also a lot of pain relief. Okay, so that's exactly what I was saying. So exogenous opioids, okay, so there's a difference between endogenous and exogenous opioids. Does anybody have an idea? Yes, go ahead.
Like exogenous would be ones that like you take like in a medicine or something.
Yeah.
Endogenous is ones that your body produces.
Exactly. And so when I'm talking about exogenous opioids, it's dealing with the things that we intake or the drugs that we intake, right? And so again, when we have opioids that are released in pain region, it induces less pain, and if we have like the reward region of the brain, then it gives you a high, right? And that's pretty much what makes people really addicted to opioids in the first place.
Can I make my comment?
Yeah.
So anytime you experience pain, you get dopamine release and endorphin release. Okay, that's why people get addicted to running, because when you're really exhausted and you're tired. So addiction is not just a bad thing. You can get addicted to good things like running or extreme exercise or anything that the body experiences as tough.
Yeah.
It will release endorphins and dopamines, and so.
Which is why, like, when you work out, right? If you work out in the morning, you will be insanely you'll feel so good afterwards, and you will be able to be really productive because of the amount of endorphins that are released after you work out. Right, so when I work out at night, I can barely sleep because I have a lot of endorphins running, and I do a lot of work, and I sleep at like 4 a.m., so it doesn't work out.
So these are really natural phenomena that occur all the time, and you don't really realize it sometimes.
Exactly. And so, so when people repeatedly use this drug, it causes certain things to happen within our receptors and also within the neurotransmitter that's released. So when you overuse a drug, it can cause dopamine receptors to become less sensitive, and so it means that you need more of the drug to cause the receptors to attach to the dopamine, right? So when you overuse it, you need more essentially to get more of the effects of the drug. And then the opioid receptors are also can be reduced. So for example, right, so these receptors, the ones in pink, can be reduced, or the opioid receptors over there in blue can be reduced in general. And so you have less binding sites for the neurotransmitter to bind to, causing it to mean that you need more to get it, right? So that's the basic idea of it. And then there's also a really interesting idea regarding your body's homogeneity. No, that's not the word.
Homeostasis.
Homeostasis, thank you. So basically, your body wants to always maintain homeostasis, right? That's the idea of life, right? And so your body wants to maintain this even line this entire time, right? So we have homeostasis right here. And let's say I take an opioid pill like at 3 p.m., right? And I'll take it at 3 p.m. And so I'll go through this idea. Let's say this is like my my blood or my heart rate. Yeah, let's do heart rate. Okay, so when I take opioids, it's going to reduce my heart rate, right? So I'm going to go downwards, and then I'm going to get that intense feeling of euphoria every day at 3 p.m. Let's say, right? So what your body is going to do to maintain this homeostasis is that it's going to automatically go up every day at 3 p.m. just so it can maintain it, right? Just so it can cancel it out. So every day, if I take opioids at 3 p.m., it'll go down. But what my body will start to do at 3 p.m. is it'll start to go up, right? So my heart rate will start increasing at 3 p.m. so that when I take the opioids, it's going to come down back to homeostasis, right? Does that make sense? And so what happens is you'll kind of get addicted to that sense every day at that certain time point, causing you to, again, get addicted, right? And then let's say like I'm at a party, right? I'm at a party and like my friend is like, yo, bro, take this, right? So I'm like, I'll take it, right? And my body isn't gonna expect me to take opioids at that time because I've been taking it like every day at 3 p.m., right? So when I take it at a different time point, it's not gonna expect it. And so the feelings of those drugs are gonna be like severely amplified. So instead of dipping down here at every day at 3 p.m., I'm going down to levels like right here, right? Which is really dangerous, and that's how overdoses happen. Because you take the normal amount that you want, but your feelings are amplified because your body's not expecting it. And so you start feeling it a lot more. Okay, so the main thing to really understand about this first part is that like OUD, so opioid use disorder, right? And so it can be people out there that can be led into opioid misuse from heritable factors. So there are some genetic factors out there that can affect opioid use disorder. It's not entirely just because you get prescribed it and you start like going crazy, right? But there are certain genetic factors where certain people are going to be more susceptible to OUD rather than others because of these genetic factors. And that's why it's really important to find out what these genetic factors are, right? So that we know which people are going to be more susceptible, and that's where we get into like genetic counseling and things like that, right?
Can I say something?
Yeah.
So there's a whole field of science about genetic predispositions to drugs called pharmacogenomics. Okay, and this is an entire area of science to understand. For example, when I take a drug, I metabolize it one way, but you may metabolize it in a different way. So the dosage I would need to be effective for me to be different from you. So there's a whole field of science dedicated to how do you dose someone, how do you know what their metabolism of the drug is going to be like. It also might be that I would have like no effect or marginal effect, and it could kill you. So there's a certain percentage of people in the population who die from drugs that would affect almost no one else. And so so those drugs are not on the market, because if it kills one person in 10 or 100,000, that's too much. So there are many effective drugs out there that are not on the market but if you could identify that one person, if you knew who not to give the drug to, it would make an enormous difference in the number of drugs that we have available to treat disease.
Yeah, and that's why they're doing this program,
Yes.
right? Okay, before I get to that, okay, so I wanted to ask you guys, so what was the main goal of this paper? What did they want to accomplish with
Okay,
this?
so
Yes, go ahead.
They wanted to establish a link between like certain polymorphisms and opioid use disorder.
Yes, right, exactly. And so, what was the main problem that came up within the first part of the study where they were like, yeah, go ahead, go ahead.
They didn't have enough patients that just were being studied for OUD.
Exactly, right? So there's not enough people out there that we can that we can use for samples to use for finding out where these genetic factors lie, right? And so, what did they do to solve that problem?
yeah.
So what they did was they did this multi-trait analysis in addition to other GWAS in order to basically enhance statistical power using a lot more groups of people so that they could get maybe find more SNPs than
Exactly.
just the population they have.
Yes, and so here's
So what's GWAS?
GWAS, right? Yes. So GWAS stands for Genome-Wide Association, right? I hope you guys remember this from 3200, right?
Yeah.
Genome-Wide Association study. Yeah, there we go. Okay. Can somebody explain to me what that is?
Basically they are trying to look where in the genome for like specific SNPs that are associated with certain disorders or diseases.
Yeah, exactly. So when we think about finding like certain genetic traits, right? So I think this is a really good example that I found out from a video. So I'll take a little bit of time to explain it because I think it's important to understand what exactly the root of the study is, right? So
Well, let's not get too deep
Okay,
in
okay.
ways because
Yeah, that's
we
fine.
need to get to the paper. But
yeah.
so just generally, it's an association between. So if you sequence the genome, you find a trait, and it's associated with people who have that, like a phenotype. Okay, and so if you sequence everyone in this room, all their genomes, and there was a gene for blonde hair, you could correlate that, with Carolyn. Okay, so that's basically the idea. Okay, is that it's a correlation between SNPs, which is the good news and the bad news, because the correlation has to be statistically
Significant.
beaten into the ground 6 ways from Sunday. That's why this paper was so much fun. Okay.
So I have another question then. So what were the ways, or what was the link that they used, or what was the thing that they used to increase their sample size? Like what attribute or what specific genetic factor did they use to find what, or to increase their sample sizes?
Carolyn, okay, we're just going around the room.
I don't know if that made sense, so let me, if you need me to clarify.
Did I torture you enough or not?
I think I'm good.
Okay. Just let me know. Okay.
Yeah, I guess I'm a little confused with what you're asking, because I guess they,
Yeah, so
Well, let her go. Go ahead.
yeah.
I guess they pull data from previous studies.
Yes, so they pulled data from previous studies, but what was the data that they used to help them find out who has more of a likeliness to have OUD? I think that's what I'm
Okay.
asking.
Yeah.
Was it when they were like looking at like alcohol use disorder?
Yes, exactly. So what is that called?
No, keep
Yeah, I'll just
What
keep
is
going
that
in
called?
order.
No, keep. What is that called?
Oh yeah, yeah.
Was that like the multi-trait analysis?
Not really. That was like the study that they did, but there's something inside of there that they personally looked at to really know what exactly they needed to do. Do you know?
Was it the Phenom-Wide Association study?
Not quite. Not quite.
No, okay. So-
I'll go this way. Pretty good. You can go ahead.
No, I thought the same with multi-trait analysis, but.
Okay.
Genetic correlation.
Exactly, yes. So it was the genetic correlation, right? And so this is an idea where we have relatedness between two certain genes, okay? And this is where our definitions come into play. So I'll come back to this. So can someone explain to me, okay, I'll go back. Can someone explain to me what linkage disequilibrium
Okay,
is?
back to you.
That's me.
Mhm.
Is like when like
You're good. You're good. It's okay.
So it's like non-random association of alleles at different sites. So like basically two different things can be like inherited together.
So what makes it non-random?
It's like recombination.
What makes it non-random?
What makes
Yeah.
non-random?
What makes the linkage non-random?
That they're closely together on the same chromosome.
Same chromosome. Okay, so things that are on the same chromosome are inherited together. So that's not the same as inheritance in a general sense. If two alleles are on the same chromosome, they're inherited together.
Exactly. So if I have, like, let's say I have a sequence, right, of C, T, A, G.
Okay, but you don't have to go into this.
Okay, I
we
won't,
got
I
it.
won't.
We got it.
Okay,
We got it.
good.
Okay.
All right.
This is great.
Yes.
You know, but-
I just want to provide background.
I know.
I love
You're
this
doing
part.
great.
Okay. Okay. All right. Let's go on to the next part. So going back to genetic correlation, right? So genetic correlation is like a ratio, pretty much, between certain genes that are related, right? So what they're doing with that AUD, right? So that's alcohol use disorder. They found out that there is relatedness between that gene that causes alcohol use disorder and OUD, right? So there's a relatedness between there. So if we do a G,
We got it.
okay, got it. Sorry. Okay.
I just want to get, I'm just going to move you along. This
Thank
is
you. I need that
wonderful.
because
Okay,
I'm gonna
but I'm
keep
just gonna
talking.
Move you along.
Perfect. That's fine.
Okay.
So okay, let's go on to linkage disequilibrium. This LDSC. Okay. Did anybody quite understand what exactly that meant? Without obviously the answer's up there, but did anybody quite understand what that meant? I'll just explain. Okay. So it allows us to calculate the genetic correlation between the specific phenotype, right? So that phenotype WAS association, right, or the WAS, right? They use pretty much LDSC to calculate the genetic correlation between certain different disorders and OUD. Okay, and then we have SNP heritability, which shows the variance explained by all SNPs in a GWAS and conventionally unrelated individuals. So there are people in the GWAS that aren't as related to each other as much, and so they basically try to explain that variance through SNP heritability. There's polygenic risk score, one way by which people can learn about the risk of developing a disease based on total number of changes related to disease. So it's pretty much what you were talking about earlier. And then the other thing, this is dealing with table 1, and I'll get to that later. A core is a group of people who have been included in a study, and a phenome is a complete set of phenotypes resulting from genetic variation. Okay, let's go to the paper.
Good.
Okay. So should I just go to the first table and we can discuss that?
So I think you've pretty much covered the introduction very well. So I think we're ready to dive into the figures.
Yeah, okay, perfect. Oops. Okay, I'm gonna
so-
zoom in a little bit. Okay.
You can eliminate.
Oh, yeah, you're right. Oh,
Yeah.
perfect, okay. Okay, I don't know where we're at,
We're
but,
right there.
okay.
Titleist.
Yes.
Our golfer.
What's your name? Govinda. Okay, can you explain to me what exactly this figure
Okay,
is?
so what question are they asking in this figure?
How they categorized the groups that they were basing the studies off of. So basically it would show you all the groups that they were talking about and how many were in each and how they branched and grouped everybody together to get the sample sizes that they wanted.
Yeah, so these are previous GWAS studies that have been done over OUD.
So,
Sorry, okay, yes, you're
I
right.
know, I love your enthusiasm.
Yeah, sorry.
I'm just gonna rein it in a little bit.
Okay.
So these are from previous biggest US studies, okay? So what are they measuring here? What are all these numbers?
In the table, these
Yeah.
are the sample sizes they had and what they chose to include in their studies.
So then, what is the EUR cases and the EUR controls and the EUR ineffective?
No, but what is BioVH?
That is one of the genome banks that they access.
And IPSYC?
That is the psychiatric group that they had in there when they were trying to base the opioid use disorder with psychiatric problems for correlation.
So does this look comprehensive to you?
Like I see the numbers, but like in the grand scheme of like why they chose those numbers,
No, not the numbers, but the group. Are the
Yes.
cohorts appropriate, you think?
Yes.
Do you agree? I think so too. Okay, and the numbers, okay, 639,000 people, is that a reasonable sample
That's
size?
a very good sample size. Yes.
Incredible.
Yeah, it's massive.
No, it's incredible. I mean, the statistical power of this makes it,
Yeah, amazing. And like, does anybody want to explain to me what the ineffective is?
okay, next.
So the ineffective, right? So that's the third column right here. What exactly did that mean?
Are those the cases that are like,
Go ahead.
people like show that certain like genes or that phenotype or genotype?
Kind of.
No, go ahead.
The ineffective is basically the sample size, and the effective means it's big enough to like run a statistical test.
Exactly. Yes, exactly. So it pretty much allows us to have a proper sample size that is almost equivalent to a simple random sample that we get, right? So it gives us a really, really good accuracy of what we're doing. That's basically what they're showing here. Can someone explain to me what the control definitions are?
Back to you.
For the guy, if you could explain like the control definition. So the unscreened and the no OUD diagnosis. What exactly did this mean?
Are those the people that are like not diagnosed with that?
So there's two terms here, right? So the no OUD diagnosis and the unscreened. So what's the first one? And then what's the second?
With the no OUD like diagnosis, like they don't have any correlation towards like, or like they have not been addicted to opioids ever.
Yes, so they don't have any affiliation towards OUD, right? And then what was the second one? Unscreened.
Like they couldn't be assessed for opioids.
Yes. So they had no idea, right? So they just didn't know. And so this part's really important because these are the things that like really matter, especially in controls, right? Yeah, go ahead.
If they had unscreened people in their control, was there a chance that people with opioid use addiction could be inside of that?
Yes, right.
Therefore, like reducing their statistic.
Yes. That's why that's important, right? Because they said some of these studies have been not published, and I'm pretty sure that the ones that are unscreened are the ones that are not published, which is like really important to know. But this is also like why they have such a huge sample size, and it's also why they were able to get a lot of samples because they chose from unpublished areas of GWAS, right? Yeah.
So if it's like unscreened and they don't know so much about it, why would like they want it like put it instead just to increase the sample size?
Yeah, I guess they really wanted to know, right? I mean, I'm not sure the answer to that.
Well, what's another, and you touched on this, what's another really important aspect of these alleles? How widespread they are in the general population. population.
Oh, yeah. Yeah.
Right?
Yeah.
Okay, so that's why they need huge numbers, okay? Because, it's like XYY. There's no phenotype to being XYY, and there's all this controversy about how general is this in the population. If it's something with no phenotype, so if you've never taken an opioid, okay, you might be addicted to running, but you're not going to be addicted to, so they wouldn't know that.
Yeah.
So we touched on that, and like what is the general occurrence in the population? That's why the control numbers have to be big,
I see. That makes sense.
right?
Yeah. Okay, I think we're good with this. Let's move on.
Okay.
Everyone, any other questions?
Yeah. Okay, I'm gonna touch on this one just a little bit. This one's.
No. We're gonna go to the next person.
Yeah. I'm not gonna say anything. I'm just saying I'm not gonna spend too much time on this one because it's.
Well, it depends on whether they can explain.
Sure. Okay. Yeah.
You're
Well, it depends
amazing.
on-
I feel like I need to rate you.
Yeah.
Not
I'm a
because,
nice
I
one.
love your enthusiasm, I love the way you're doing this, okay? I just need to keep us on task.
Yeah.
So.
No, wait.
Wait, I want first.
About the last week.
Oh, okay. Yeah. Go ahead.
So the purpose of the study would be like you could screen somebody, and if they're at risk for opioid use disorder, then you would be less likely to prescribe opioids to them because their chance of getting addicted is greater. Is that true?
That's one application of it, I'd say, right? Like, I think that's one huge reason why they're doing it, but it's also because of like knowledge, right? I think that's.
No, I think that's exactly
Okay,
why they're
never mind.
doing this, and that's the most
Okay.
important reason. I know we all want knowledge, but that's the reason. Because if you can identify people in the population who have these susceptibility traits, you don't give them an opioid. You shouldn't give anybody an opioid, generally, okay? But that would be an indicator, don't do this, and that would save people's lives. Okay, sorry.
Okay. Yeah. Go ahead.
Okay, so this is basically just a schematic representation of some of the data that's in table 1, but then it also answers the question, where did the data come from, showing the 7 different cohorts, I guess, they pulled data from, and also, is there an association with other substance abuse disorders, which is like the green and the red is looking at the association with cannabis use disorder and alcohol use disorder, and those, like the RG equals, those numbers are the genetic correlations, so that's how much those are associated with the OUD, and then it has the MTAG, which is kind of showing that area is like cannabis use and alcohol use are combining that with the OUD to do multi-trait analysis to get more.
Exactly. Yes. Nailed it. Perfect.
Excellent, yes.
Yeah. That's pretty much all this research showed. You pretty much covered every part. I don't really know. And then like they have the ineffectives right here, right? I guess that's also pretty important. All right. Perfect. Cool. She just did my job for me. That's great. Okay, this is probably my favorite figure. I really like this figure.
Who's your favorite too?
No, I like these. I prefer this. I just felt that there was stuff missing, and what they were also saying in the paper. I was like, where is it? I can't
Okay,
find
we'll
it.
get to that.
Yeah,
Okay.
I agree with that.
Arisha.
Alright, so these are basically the Manhattan plots, and so like in figure A.
No, so what are they asking?
Oh, sorry. They're asking like which like alleles are associated with substance use disorder. So
And how are they measuring that? And how are they doing that?
they're looking at like the different polymorphisms that are like more present in people that have substance use. Yeah.
Exactly.
Correlated.
Yeah,
With substance use disorder. So then, should I go on to explain? Okay, so in A, we see they're looking at European ancestry GWAS results for opioid use disorder, and we see that like the OPRM1 and furin are like above the like
Threshold.
I don't know, yeah, the threshold. So those are like more correlated with opioid use disorder, and you can also see like which chromosomes those are on. And on B, we see like that's from, I think it's from the multi-trait analysis.
Yes.
And we see like a lot more like associated
Why do we see a lot more?
Because like I think it was just because you had a lot more like your sample size was larger and you're also looking at like more grades.
Disorders, yeah.
More
exactly,
disorders.
Exactly.
So like these are not just associated with opioid use disorder,
Exactly.
it's like with other
Exactly.
alcohol use.
Yes.
Other substances.
So what's OPMR?
So is this
Don't knock you.
I'm not saying it. It's just gene right here.
Yeah, what is it?
OPRM is one of the approaches that we want to see if there has any correlation.
No, but what is it? What is it? What gene? What does it encode? What is the gene code? What is the gene code?
This is huge. This one's like probably the biggest part of the paper.
It's only the most important part of the paper, but hey,
It encodes a mu opioid receptor.
Yes, yes.
and keep going. Tell us more about it.
I think those are what are like
It's the opioid receptor.
right, yeah, that's I think when I looked it up it said something about it was like a proteolytic enzyme or something that like breaks down and then
Yes.
that breaks down other proteins in the brain and the neurons.
Yes, yes, exactly. So it's
What's that called in biochemistry speak? Turnover. Turnover. So this is responsible for turning over these things so that they don't stick around forever, right? Okay, so turnover is really important in the regulation of how a protein will affect you. If it's long, the longer it sticks around, the less you have to make. So turnover is a key in regulating these responses. Okay.
Okay, I have another question. So, you told me earlier that there was significance in, I'll ask this. What's surprising about this figure? What's the most, like, the biggest thing that you can take away from this?
Okay.
The biggest thing that you can take away from it is that there are many different, that they uncovered many different, I guess, alleles that correlate to this disorder.
Not what I'm looking for. Yes.
I was surprised when the OPRM one dropped below the threshold, and yet throughout the whole paper they're like, OPRM one is so great, and then now you're looking at it with all this extra data, and you're like, oh, it's not so great.
Exactly. Okay, why? Why do you think this happened?
Okay, go ahead.
Well, I have a question. How do they calculate that bar for significance?
The what?
The threshold. How do they get where the threshold would be?
Oh, I wish I could tell you that. I don't know.
I thought I read something about like an N effective of like 80% or something. Like
Oh, you're right. Okay, yeah.
I can't go any more than that.
That's good enough.
Yeah, I think that was it. You're right. But yeah, that's how they did it. So going back to the question, why do you think this gene that they were so certain about that was gonna code for OUD, and when they do this and I forgot multi-trait analysis, it's not even above the threshold. Why do you guys think that is? Yes.
They talked about like the cost of specificity when you increase the sample size.
Exactly. So it's because we have so much going on, it can cause our like sample size or like our whole entire analysis for the GWAS to kind of go through the roof because of how many samples and how many things that we have. So it reduces the specificity of what we're actually trying to look for, and that was one of the consequences of having a huge sample size, and they got that consequence.
Does that make sense to you?
I mean, kind of, yes, to a certain extent, because like.
So the bigger the sample size, the less. The
Oh,
bigger the
I see,
sample
I see.
size, the less statistical significance you have. Is that what you're saying here?
I see what you're saying. Okay, it doesn't make any sense at all. Then, yeah.
The only reason I'd say it could potentially make sense is because the first one was European, and what if now, because they're pulling from so many samples, they're getting the cross ancestry to different people. So what if this OPRM1 is actually very significant in European populations versus other populations? And that's the downfall of GWAS when we have so much European data and not other populations.
Yeah, I think you nailed it.
Absolutely perfect. Okay, so what do we in first world health? What do we measure? Do we measure European allelic, variation? It's all European. Okay, what do we know about allelic variation in Africa?
It's
Zero.
Yeah, it's
Okay,
really bad.
well, in Asia, well, apart from, major countries even in India, what do we know about any of these kinds of things? Huge populations of people that are completely unstudied. Okay, so that's exactly right, and that's why it drops down, because we don't know. And so, does that make their claims about this gene less significant to you?
I wouldn't say so.
What do you say?
I would say no, just because you have to understand the parameters that go into each experiment. If you're looking at both of them, just assuming they're telling you the same thing, then it doesn't really make sense. But you have to take into account the data that they're pulling from.
So I love this figure because it tells you something really important to keep in mind, that you can't just look at this at face value. You have to dig a little bit deeper, like, okay, they were looking at European populations. You have to be able to drill down into these data and dissect it and figure out exactly, why these things are happening. But because when this goes down below the level of threshold, you go, what?
Yeah, exactly. All right.
Okay, good.
Okay, this figure. Oh my gosh.
Whose turn is it? Carolyn, you haven't done. Did I skip you? I know it's your turn again. Okay, what are they doing? What question are they asking here?
So I guess they're just showing,
No, what are they asking?
what markers were, I guess it's kind of like the table that's associated with the Manhattan plots. It showed the markers, I guess we ran statistical.
I can't hear you. You could just put your mask down and shout.
I know I just had COVID last week, so that's.
Oh, that's right. Okay, so, yes. But you've tested negative since you had it, right?
Yeah, I just thought that was.
I know, I appreciate that. I totally
okay.
appreciate that. Okay, don't pull your mask down.
Yeah, so they were just showing, the markers that were most significant, to in the European population in A, and then B is the cross ancestry analysis.
Okay, so let's start like on the left-hand side, right? So what's that showing? Like, clear left-hand side. Chromosomes.
Yeah.
And let's go down a little bit more. What are the markers?
I guess the markers they looked at in the GWAS.
Inside of what? Can somebody answer that?
Those markers are the SNPs located inside those genes. Like the majority of it is OPR and one, and then there's one furin S SNP that they found, and then they were looking at how significant was it.
Yes, exactly. So what are these p-values? What does that mean? What does it mean to be statistically significant in this
Okay,
context?
Carolyn, what's the p-value?
It's a statistical threshold.
Yeah.
And what's the threshold? What is a p-value? What does it have to be significant?
Usually 0.05.
But in this case, what are they using? Which threshold did they use?
Okay.
Huh? I don't know.
I thought
Wasn't that?
for a GWAS it had to be 10 to at least the -8
Yes.
in order for it to be significant.
Yes. So you see, like, these p-values are like really, really low. Like, and so what they're doing is like what are they doing with that? Why do they have such a low threshold? Yeah.
They're using the bond for early correction to increase there's a lot more false positives or false negatives when you use the 0.05, so you have to correct for all the
Exactly.
data.
Yes. And
So
so
if you're gonna look for a needle in a haystack, it can't be 0.05. Okay, it has to be incredibly better than
So they're being really, really hard on the setting, which is one thing I really liked about it, because like you can tell how much they want to find out about this just by the amount of effort that they're putting into the study. Let's see. So can someone tell me about what this right-hand side of the table shows, the African part?
that. Okay.
Shows the p-value for the African ancestry.
And so what are those p-values showing you? Like, what do those mean?
The p-values are greater than, no, are still less than the threshold.
You were right the first time. So yes, so they're
Say it
great.
again.
They're greater than the threshold.
Yeah,
And what does
exactly.
that mean?
It means that what they're, it means that it's standing out when what they're looking for or what they're testing is proven to be true.
I'd say the opposite of that, right?
Yeah,
So,
it's the opposite of that. Okay, it's the opposite of what you said.
yeah.
Okay, so when a p-value is greater than the threshold, it's not statistically significant.
And so that's what they were saying pretty
Okay,
much.
just repeat
Yeah, okay, sorry.
So it's not statistically significant, so that's really important to understand, okay? That a p-value tells you what it has to be for you to believe it, right? Okay.
Yeah, and so in the paper they were saying like the data from the African side, or the data that the GWAS that they got from the Africans, right, or the Africa, right, was not used because it was like you can see it from the data, like they're just not significant, so we just didn't even use that. So yeah.
Okay.
Yeah, go ahead.
Can you explain the direction?
I wish you wouldn't have asked that. I have no idea. I tried searching it. I literally had zero idea. I genuinely did not know.
Yeah, question.
Do you understand it?
The direction? Absolutely not. I couldn't figure out what they meant by like the question marks.
Yeah.
They went with pluses and minuses.
That was probably the most confusing part of this week.
Okay, I asked a population biologist in our department and they didn't know either.
Really?
Okay, so.
Okay, good.
Okay, so
Thank-
I asked, I won't mention the person's name, but
Yeah.
this person is a world-class statistician. And he had no idea.
Okay, well,
So-
that makes me feel a little better.
So I think, just so you know.
Okay, alright.
Okay.
Cool. That's great.
So what does that mean? What does that mean? Should you trust this because they said so? No. If they don't justify their analysis and they make it so impenetrable that you can't find it out, dismiss it.
I agree. Okay.
I would hesitate to say they did that on purpose, but who knows?
Ooh, this one's really good too. Okay.
Okay, yeah, this is good. Okay, so whose turn is it? Oh, back to you.
Yeah.
Oh no, let's do it through your side. Okay.
Okay, there we go.
I'll try to be equally mean to everybody.
So I think for this table, what they did was they
What
just,
question are they asking?
how correlated is the results of this genomic association with other like negative health? Well, not just negative health outcomes, but also things like educational, how much education you have,
Yeah.
or how much exercise you do, and things like that.
Yeah.
So.
Yeah, so it's a really interesting figure. So,
No, that was good.
yeah, that was good. I'm trying to find anything else, but.
No, that was good. So yeah, they want to put this into context of the real world, right? Okay.
And so, which ones are?
So go ahead and explain what they found.
So obviously, opioid use is going to be correlated with opioid
Yeah.
Generally, the more negative, like things that you would expect to be addictive disorders can be pretty strongly correlated with opioid use disorders, but also other things that sort of make sense, like having a higher level of education and less just pain in general, better income. Those tend to mean that you're less likely to be susceptible to opioid use disorder or be correlated with opioid use.
Does anything on this list surprise you?
Yeah, I was just about to ask that. Which was the most surprising thing? I'll ask you the same question.
No, anybody.
Yeah.
What surprised you on this
I was
list?
like, finally, I just thought it was really interesting that they put, like, the age of first sexual intercourse on there.
Yeah, that's what I found most surprising.
Was what?
That was crazy. This last one. Crazy. I did not expect that. But like, I guess they put it there. So, well, what does that mean? What does that mean?
It's like really negatively correlated with opioid use disorder.
Yeah. So the less age, like the lower age that you lose your virginity, you have a higher risk of opioid use addiction, which is it's kind of crazy, right? That's
We
not.
become addicted to endorphins sooner.
Yeah, I guess so. That's probably what it is, honestly, right? But yeah, that was probably the most surprising. I thought that was really interesting. Did anybody else want to share what they thought that was so surprising?
You don't have to tell us what age you are. Okay.
Yeah, go
I was
ahead.
gonna say another thing that was interesting is like a lot of it, the higher ones were not just like drug use, but it was also pain outcomes. So like, a lot of it is just like people who are like, it's like I mean like from like I have a friend who has sickle cell, so he has to take opioids for like pain.
Yeah.
And like he tells me like he like makes sure not to like use that whenever.
Yeah.
But it's just interesting to see like how like people like who are going through like stress and pain have to kind of rely on this to.
Exactly. Yeah. It was a really good thought. Yeah, go ahead. Go
I
ahead.
thought it was funny about how they included body mass index because like
Yeah.
the dosing of drugs based on your body weight. So like technically, if you wanted to get like more euphoria out of it, you would have to like exponentially increase your dosing from the start point.
Yeah.
So you'd be less likely to probably go broke.
Yeah.
So like it makes sense.
Yeah. I like that. Yeah.
I think what's interesting is like the taking other prescription medicine because I think in news and stuff they put like opioid addiction and like prescription medicine addiction like very much hand in hand and like that correlation is not as big as we we think as the public and society. So I thought that was really cool.
Yeah. Yeah, I like that too. I also thought this one was really like last month headache. Like if you got a headache last month, then you have a higher chance of obesity. I don't know. It's like.
There's also a strong genetic component to obesity. I don't know if you understand it, but people, there's a receptor in their brain. They always are hungry, and no matter how much you eat, you're still hungry. And we did a paper on that a couple of years ago, and it was just sad to always be hungry.
That's crazy.
Yeah.
Okay.
Okay. I think this is so cool.
I thought this was. I think this was the
it's
coolest
so
one.
cool, you know. And people who are on other meds probably because they have pain, that they have other illnesses and they may not be a pain reliever, but they have other illnesses that are causing them pain.
Yeah, and so like that's why the last one headache. Like we normally get headaches like here and there, right? But people who are actually prone to getting headaches, it makes sense, right? Everything that they put here makes perfect sense. Okay, I think this is the last figure.
Okay.
All right, this one's also a doozy.
So let's see.
Okay.
Okay. What question are they asking this?
They're asking what SNPs are of significance for the multi-trait study,
Yeah.
and then they're also looking specifically at the cohorts for the alcohol and for cannabis.
Yes. And so,
There's a whole institute in the National Institutes of Health. There's one for general medical sciences. There's one allergy and infection disease. There's a whole institute devoted to addiction to alcohol and the genetic basis for addiction. And some of this work comes into that, but there's a devoted to understanding the genetic basis of addiction, which is cool, good.
Yes. So which genes or which markers were significant? So are like how many were significant in AUD and how many were significant in cannabis?
So in the AUD, I'm pretty sure there's the ones that you bolded.
Yes.
You see 3 in AUD, and
Yes.
you see, there's only 1 in cannabis.
Exactly. So what does that mean?
It means that the markers they found in MTAG can be correlated. Which I'm not so sure.
I'm not sure about this either. I'm asking you.
I
Like, I'm genuinely asking you. I have no
I
idea.
think it's either way, where it's like either more of the traits, the SNPs that they found are correlated with opioids, or they're not correlated as much with any of these three. It could be something else.
Yeah. So
It's like I'm not really sure which way to read it.
So what I thought was, and correct me if I'm wrong, this is the one question I have for you, is it that whichever ones are significant are actually correlating to more OUD? So like the genes that are present here, the markers that are present in people here in AUD have a higher chance of also having OUD. Is that what they're trying to say? Okay.
Because addiction is addiction.
Yes.
Okay, and so what they're trying, and one of the surprising things about this paper is that there is this correlation, that there's a general pattern emerging genetically about what constitutes a predisposition to susceptibility to addiction.
So then the bolded ones are saying that like those with the opioid use addiction could likely be higher risk for also having these other ones.
Yes, and that's really important.
Yeah.
So you don't have to do a genetic screen for everything.
Yeah.
If you could just use one indicator, and that would cover a lot of territory. Okay, then you can diagnose a predisposition by looking at just a few alleles, if you can know how they're correlated to each other.
That's really interesting. That's really cool. Okay.
I thought it was interesting. Did
Yeah.
you love this paper?
Yeah, I honestly, okay, it was a headache, but like.
Okay, I have to take ownership because someone didn't turn in their papers on time.
My
So I
bad.
chose one for him, and I thought, what greater punishment can you imagine for someone not turning in their papers on time than to assign this paper?
Yeah, and dude, I spent so much time on this.
I
spent a whole lot of time.
But I also like the paper because I think there's a lot of really cool stuff in here.
Yeah, and I think it's really cool that there's like a lot of application to it, right? Like, think about it like when you guys, I know some of you guys want to become doctors, like think how this applies. Like, this is huge, right? Like, this is a huge finding. Yeah, go ahead.
I just have a question about this one. So for allele 1 and 2, all the nucleotides for allele 1 are A and T's, and then for allele 2 they're all G's and C's. What is that like allele 1 is like the normal nucleotide for the gene, and allele 2 is the SNP?
That's a great question. I thought it was like changing from a T to a C, but now that I'm looking at it a bit more, it's probably not that. I have no idea.
The wild type is an allele too.
Right. Okay, so is allele 1 of the wild type?
That's what I got. That's what I thought.
Okay.
Okay.
I just thought it was interesting how they were all A's and T's and they all switched to G's and C's.
Yeah.
In my point of figures that you went through in the chart, it was that chart on this page. So the one for
This one?
yeah, the one for urine, above it. The one for
This one?
that one. The one for furin, it says allele 2 is T's. Like it's an addition, while the other ones are a substitution. Which I thought was kind of cool because like that would produce a friendship. So that would be like, what else does that do in their body? Do they know that?
So then would this be an insertion then? So this A2 would be an insertion just first one because they're adding a nucleotide as well as keeping the old one, right?
Right.
So then it frame shifts, and that's probably why it's so significant, right? Okay, got it.
Exactly.
Makes sense.
And
Good
there's something
observation.
even more subtle about this, when you have GC-AT base pair changes, or AT and GC base pair changes, what that's possibly telling you is that it's in the stem loop structure of the protein, and that you're disrupting, or in the RNA, that you're destabilizing some structure, which would be suggestive of an experiment to figure that out, so you'd model it.
Okay, I think we're good with this.
I think we're good
Right?
with this.
All right.
Okay.
All right, let's go to the social paper then.
Absolutely.
Right? Okay. I'm not gonna bring it up, to be honest, because I don't think we need to.
I know, we can just talk.
Yeah, we can just kind of talk about it. So, I guess I don't know where we are, but I guess what was the one thing that you guys found out from that paper? I don't want this to become like a political discussion at all.
It can be.
Sure, okay,
Why
fine.
not? We're
Yeah,
all
we can do
adults
whatever.
here.
Yeah, sure.
We're all respectful of each other's opinions.
Yeah.
And so the point of this is, say what you think, as long as and it's okay to disagree.
Yeah.
It's okay to bring up whatever you think, and to me, the most important thing is civil discourse. And you learn more from people who don't agree with you than you do
Exactly.
from people who do agree with you.
Exactly.
So don't hold back.
My biggest thing is that there's two sides to the issue. There's a reason why. So, yeah.
So when I was shadowing the neurologist, we had a debate on this, and the Supreme Court ruled that doctors that run their own practice are a business, not a commodity, and so that's why they are able to raise their prices. Because insurance will only give you this much, but they need to make this much to make ends meet, so they have to raise their price. And now insurance raises their price, so it's like an ongoing battle.
Yeah.
And
So let me ask you, is healthcare in this country a right or a commodity? How many do you think is a commodity?
What we think it
I'm not
should be
saying
or what it is?
what it should be. We
my
can
bad.
all agree, I think we can all agree that it's now a commodity,
Yes.
okay? It's for sale, okay? The other way to look at this is that it's a right, which means that changes everything. I mean, that changes your whole, to me, it changes your whole political view about how healthcare should be supervised and regulated in this country, okay? And so that gets to your point. If you're a physician, you can charge whatever you damn well please. Okay, and if you're a drug company, you can, till very recently, you can charge whatever you want.
that's how they do it, the pharmaceutical companies and the insurance companies. The insurance won't pay this much, and so they keep raising their price because now they have to cover more. And so it inevitably comes out to just paying more and more progressively as the years come out.
Yep.
And then this paper touched on all those patents. Those patents are really bad for the medical industry because they can literally just change the capsule that they're in, and that's another 20-year patent on it that no other company can access, and they can charge
But is there
whatever they
any
want.
good side to patents?
No, I don't see it because a lot
don't
of it
see a
was
good side
public
to
knowledge.
patents?
I think there is, because if it's like a small company that comes with like a revolutionary breakthrough, like a lot of times a big company will come in and steal it, but a patent prevents that.
Yeah.
Well, but what about recouping the money you spend on research and development?
Yeah.
Why, patents only last 7 years. Okay, so for 7 years, so you go through clinical trials, yada, 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 yada. You spend a lot of money on research and development. You spend a lot of money on clinical trials. You have to get USDA approval, and then the reason you have a patent, it only lasts 7 years. It's not forever. Okay, in those 7 years, you charge a lot of money for these drugs to recoup the investment that you made. Okay, and then they go off patent, and then there are a million generic forms of the drug that are incredibly much cheaper. But if there were no patents, there'd be no way for companies to recoup their investment. And do you think that would impact their willingness to invest?
Yes.
They wouldn't want to be the first to find the new drug anymore. They would just wait for someone else to potentially.
Exactly. There'd be no reason to be first. There'd be a lot of advantage to being second. Okay, so that's real. I mean, that's real. Patent tax allow companies to recoup their investment. And they're not a nonprofit. You know, Eli Lilly and Pfizer and GlaxoSmithKline are not nonprofits, okay? And they wouldn't be in business, and they wouldn't have the money, the backers, the people who buy stock because they want to make money. So as long as healthcare is a commodity, that's the way the world works, right?
And I want to ask you guys this, like, how does this relate back to the paper that we read? Like, people who do suffer from OUD, how does that work? Yeah.
Well, the doctors get paid more if they're prescribing more medicine, so if they're prescribing more opiates, they get a, I don't know if it's a tax break, but they get some kind of revenue from the pharmaceutical.
Yeah, they get a nice tax. They get a nice check, right? Like,
Yeah, but who made the most money?
pharmaceutical.
Which pharmaceutical company?
Like big pharma.
No, which pharmaceutical company? Unless you've been under a rock, you've heard of Purdue Pharma, right?
I have never heard of it.
You've never heard of Purdue Pharma? You should really get out more.
Yeah, I really should.
Tell us about Purdue Pharma.
I believe they're getting under like a big investigation right now they're the ones who kind of potentiated this whole opioid epidemic, and they're the ones who like pushed all the doctors to start prescribing these and using them when they really don't need to, and they're responsible for a lot of the addiction deaths.
And who owns Purdue Pharma? The Sackler family? The Sackler family? There was a wing at the Metropolitan Museum of Art called the Sackler Wing. They took the name off. They took the name off. Okay. So you've never, is this news to you?
Yeah, that's crazy. I did not know that.
So the Sackler, go ahead.
I was just saying, I learned about that in a cultural diversity of American art class, not even science class.
Yes.
Very interesting.
So the Sackler family made billions of dollars on false advertising. They told physicians that this was a miracle drug that was not addictive. They made a big deal about the fact that you could prescribe this for chronic pain, that you could take it over a long period of time, and it was not addictive. And they knew better.
That's crazy.
How can you not know that? Did you not read? Did you not read the news?
I told you,
I mean, do you not watch CNN?
I literally decided I want to become a doctor like 3 weeks ago.
So you lived under a rock before that, and that was your excuse? I mean, how can you not know this? This has been in the news for what, 2 years?
That's crazy. I didn't know.
Yeah, so you learned about this art class that took their name off a wing at the National Gallery.
And they sponsored a lot of other exhibits and other art museums around the country and in Europe, and they're all like in the process of being taken down.
They were philanthropists of the first order in art, and now their name's being taken off all these museums because and they're being sued by billions. But they've protected their money in offshore accounts. So there's billions and billions of dollars that they've sheltered from these lawsuits.
Wow.
So don't blame doctors about this. We can blame doctors for a lot of things, okay? We can blame physicians for more money and prescription, but this is not the fault of physicians. This is the fault of false advertising by greedy people who own a pharmaceutical company and flooded the market with these drugs because they claimed they were not addictive.
That's crazy. Wow.
How many of you didn't know that?
I have a question. So, like, I've heard about this, but not, like, in great detail. So I was wondering, like, you said they presented it as a miracle drug. So, like, by that, do you mean, did they present, like, false data and, like,
Yes.
try to, like, prove that it's
Yes.
not addictive?
Yes.
Oh,
Yes.
That's
Yes.
nuts.
They released clinical trials that favored their claim that it was not addictive. So who else can we blame for this?
People who peer review their research.
No. What is a government agency we could say? The FDA. I mean, give me a break. who's supposed to protect us? I'm okay, I'm taking over now.
No, you're right. No, you got it.
Since you've
I have no
been living
idea.
under a rock, I don't know what rock there is so I can provide internet service. Okay, so, the FDA did not do their job. They did not do their job. They didn't peer review these studies. They didn't look at the primary data. They let all this pass, and it went on for years and years and years before it became a public health crisis.
Was this through the lobbying?
No, it was just bad government oversight. I know that I'm bursting your bubble here by telling you that the government is incompetent at a lot of things, and that pharmaceutical companies will lie to make money, and there's no Santa Claus.
Yeah.
As long as I'm, bursting your illusions here. I thought this was what you were gonna hit on big time.
Oh, well, I probably should have. Shoot.
But what else? I mean, yeah, this was major scandal.
Yeah. I'll read on it. A bit
No
more.
kidding.
Yeah.
I'm gonna put the internet to your maybe you can put up a little antenna.
Yeah, I'll try to. Like Patrick, right?
So what other comments about this? I thought this was, yeah.
It's just more like anecdotal, but I have a friend who has type 1 diabetes and she's had it for a very long time. And last year, her insurance decided they were no longer going to cover her insulin. So she had no insulin and was rationing it for so long because it's $300 for this much, and that lasts you a very short amount of time. And she was like, I'm literally going to die. I don't know I can't pay $300 every week for, I don't know how often, probably more than that.
Being able to renegotiate drug prices
Yeah,
is gonna save people's lives.
yeah, I just think it's ridiculous, she literally needs that insulin to be alive, and her insurance just doesn't cover it anymore, now she has to pay out of pocket for it.
So should drug companies be responsible for that? I mean, should they? I mean, they're a company. Where do we stop regulating companies? I mean, when do we say, okay, you're a for-profit organization, but you have to be a philanthropist? Is that fair?
I think it comes back to the ethics of it, there should be some ethical regulation, if you're doing something that's causing people to, the direct cause of what you're doing is causing people to get very sick or die, then that.
But this is capitalism. Isn't that what capitalism is all about?
Yep.
I just, my job would be to play devil's advocate, so
Maybe they're trying to put into play natural selection.
You may be onto something.
Yeah, well, let's not go there. But one, like the paper we talked about last week, it's gonna drive, if you can, alleles that protect you from HIV that's gonna drive the population forward into having those alleles, because people who have those alleles will survive as long as they have